Hello and welcome. My name's Marc Tennant and I'm going to be talking about cells. Today I just want to get started with just the basics of the principles around cells. And please remember, cells are what a human is made of. These are the building blocks of life as we know it. We are made of a lot of cells. They are relatively small in most cases. Just to give you an example, the human brain, which weighs under 1.5 kilos, has about 1 billion neurons in it. Neurons are the special cells that actually do the electrical computations that is human life. But remember, even in that 1.5 kilos, there's another 1,000 times that number in supporting cells. Cells are innumerous in your human body. And do also remember that cells are continuously growing and turning over. In this little infant, newborn infant, I want to remind you that from conception to birth, 50,000 of those neurons were born every single second during that period. This is an astonishingly rapid process. And cell birth doesn't finish when you stop growing. Right in you today, right now, cells are being born and other cells are dying. For example, in the main artery in your chest, by the time I finish this lecture, the lining of that artery will have been completely replaced by new cells, and that happens continuously through life. And the reality of cellular biology is that it's about chemistry, organic chemistry. This is about various chemicals from the external environment interacting and producing other chemicals inside the cells that then act to do something. Take, for example, light. Light enters through your pupil, the black area in your eye, heads towards the cells on the back of your retina, and those cells convert those incoming photons into chemical energy. That chemical energy is then converted into electrical energy, and that electrical energy is passed down neurons to your brain, where you sense the information that is the incoming photons. This is a story of chemistry. And cells themselves come in a whole myriad of shapes and sizes. There are cells in crayfish, for example, of a size that you can see with the naked eye. There are cells that migrate around and can walk fundamentally to move and destroy foreign materials when they enter the body. Cells come in amazing variation. Here we have two types of cells. Well, one on the right I'm not so sure I could possibly really call a cell because it doesn't have one of the fundamentals that are a cell, but these are red blood cells. Whereas on the left you're looking at a series of neurons, two neurons, with their intracellular skeleton highlighted in yellow and green. So these are cells of all sorts of varieties of shapes and sizes. And then there's astonishing arrangements of cells. Cells are organised into clusters to do particular jobs. What you're looking at here is actually some dyeing of different cell types in a tadpole. On the right you can see the two orbits to the left and right, the red area of neurons in the brain, and you can see all the interactions between the different cell types stained in different colours in this tadpole. Cells have inside them a skeleton. This skeleton is made of various proteins, and the skeleton provides a cell with its shape and allows a cell to move. Here you can see the different skeletal elements inside two cells. The one to the left, you can see the different types of protein, actin being in green and microtubule being in red, that are the different skeletal components to give a cell its shape. And in the picture on the right-hand side, you can see the core nucleus of a cell, and we'll talk more about that later. And here's a different way to look at cells. This is the most common way we look at cells, where we look at a very thin slice through a piece of tissue. On the extreme left, you're actually looking at a piece of skin, and you can see across the top all the layers of squamous or flat egg-shaped cells laid one on top of the other to make a surface coating. Whereas in the middle picture, you can see cells arranged as cubes. These are called cuboidal cells. And to the right-hand side, you can see long, tall cells with their nucleus in the middle, and these are called cuboidal cells. So cells are defined by their shape. So when we talk about cells, we might talk about cuboidal cells, or we might talk about columnar cells, or we might talk about squamous cells. We also talk about them by their arrangement, how they are single layer or multi-layer. For example, the picture to the left we would call stratified, because we can see that it has multiple layers of these epithelial cells laid down on the surface. The other important thing to remember when we talk in general about cells is scale. When we look at things, we need to understand how large they are. And you know, as physicists, the ideal way to ensure we understand scale is to measure cells. And we measure cells in all sorts of ways. I always share this picture to try and trick people, but maybe you know what these two photographs are. Neither has a scale bar. On the right-hand side, you are looking at the surface of a planet. That's fine. You can probably calculate the sizes because you sort of know how big pieces of equipment and things are. But then when you look at something you don't know about, such as on the left-hand side here, you have no idea of the relative scale. Well, in fact, I think you probably do know what this is. This is the red spot on the surface of Saturn. This dot, or this red spot, is actually the equivalent of 4 Earths across. But because we have no scale bar, we're in danger of misinterpreting the size. So when we look at cells in bodies, it's very important that you look for a scale bar and understand the relative size of cells. And I don't need to tell you that there are different measurement scales that we use. The vast majority of scaling that we do, we look at things in the micrometer range. In other words, they're the thousandths of a millimetre or 1/1,000,000th of a metre. So that is the common scale that you'll see most of the time when we look at cells. Sometimes we look at cells at the nano and angstrom level, where we can start to see the actual molecular makeup or the chemistry in cells. And here's some examples of what micrometers look like. We can see an image of an ant here and a scale of about 20 micrometers across that scale. And I always remember that the ball in a biro is about 1,000 micrometers across. And here again at the bottom you can see our friends the red cells, which we should remember are 8 micrometers across. In humans there is very little variation in red cells, so when you see red cells in images of tissues, you can use them like mini rulers to get some idea of the scale of an image. To look at cells, we use different types of microscopes. These give us different scales by which we can magnify. A common light microscope will magnify tissues up to 1,000 times, whereas a more complex electron microscope will scale up to 100,000 times. What these two microscopes have in common, though, is that they are transmission. In other words, you need to cut a very thin slice of a tissue or cells, and then the light for a light microscope and electrons for an electron microscope pass through the tissue and you visualize it from the other side. So these are called transmission microscopes. Alternatively, this is a scanning electron microscope. What this does is use a beam of electrons that bounce off the surface of tissues that then gives you like a drone flight across the surface of tissues. And the picture you're looking at on the left here is actually the surface of a tongue, and you can see all the rough or furry projections from that surface, pseudo-coloured in red, from the surface of a human tongue. So you can see that a scanning electron microscope is different from the other two microscopes in that you are able to see three-dimensional structures looking at their surface. And if you go back and look at that picture of the ant in a couple of slides ago, that also was done with a scanning electron microscope. And the fourth and last type of microscope is a relatively new one that can not only look at surfaces, it's a scanning microscope as well, but it can move molecules around on that surface. Clearly this magnifies to a much greater extent, up to the hundreds of millions of times. If you look carefully at the picture that this scanning tunneling electron microscope has made, you can work out who invented this. They have used individual atoms, and I've forgotten what the chemical is, but the blue are individual atoms that they've used a scanning tunneling microscope to move around on a surface of carbon to write the letters I, B, M. So a scanning tunneling microscope allows you to look to much higher magnifications and also actually move things around in the tissue. But the vast majority of times we look at light microscopic pictures of tissues, of cells. And here you go, 3 different cells, all looking with light microscope. The black in each of these are the nuclei in the core of a cell. And we'll talk more about nuclei in a minute. And here's a transmission electron microscope view of a cell. This is a relatively common view as well. This is much higher magnification. You can now see that those black dots have actually got some content as a nucleus down at the bottom left-hand corner there. And I've labelled up 2 more of the intracellular components that provide a cell with its activity, mitochondria and rough endoplasmic reticulum, that in the next talk we will talk about. So far I've explained cells, but please remember cells are just one level of scale when we talk about a human body. And here we can see the transition from cells on the lower right through to tissues on the upper right. We're extracting some blood or some, I think we're actually extracting some air from a lung there. And then on the left-hand side, you're seeing an entire organ. This is a liver about to be transplanted as an organ transplant. So you can see the difference between cells, tissues and organs is really a factor of size or scale. Also remember, you run into real risk when you look at transmission views, such as the common light microscope, because you are looking at a slice of a three-dimensional structure. So you are only looking at a sample of a cell. You aren't looking at all of it in its operational state. So there is risk looking at different types of images that you may miss some information or get confused. So that's where I'm going to stop with this little introduction piece to cells. Next, what I'm going to do is talk about what you find inside a cell and what it's made of.